Hello class, today we're talking about topic 3, blood vessels, and we'll be continuing on in this lecture with the lymphatic system. So structure and function, of course we're continuing off the heart. The vessels themselves will actually both begin and end at the heart. This just makes it a closed circuit, but it's actually working in conjunction with the lymphatic system to circulate the fluids of our various tissues. So in general, an artery carries blood away from a heart, while a vein carries blood towards a heart. Now we did see sort of an exception with the coronary circuit, where coronary arteries are feeding those big muscles of the heart, whereas the coronary veins are sort of draining the big muscles of the heart. But in general, arteries move away, veins move towards, and when we look at their actual anatomy, we will see some differences because of this. Now you also have these capillaries, and the capillaries are the portion of this circulation that is actually in contact with the tissue cells, and so this is where nutrient and gas exchange are taking place. So just starting at the heart, let's say we've completed one cardiac cycle and we've pumped. What's going to happen? Well, from the left ventricle we're moving out into the aorta, into the elastic arteries, the conducting arteries, moving down into the muscular arteries, which are distributing arteries. So this is where vasoconstriction, vasodilation can take place. Then you have arterioles, or resistance vessels, culminating in a terminal arteriole that leads into the capillaries. So you do need to control the pressure moving into these capillaries because they are so thin. If you slug it with high blood pressure, you're just going to lyse that capillary and burst it open. So we mentioned before that there are anastomies, these sort of junctions located along this vasculature system and even in the coronary circuit, as we saw before, but these are simply junctions. Continuing on, so the capillaries themselves serve as the exchange vessels of nutrients and gas to the tissues. From there, the deoxygenated blood will move to the postcapillary venule through the small veins, which only have the job of capacitance. And so veins just serve for storage, but as a result of that, they can also be larger to accommodate higher volumes. Now, you notice this little green piece coming off as well, and this is corresponding to the lymph vessels. And so the lymphatic system is essentially draining excess fluid from the tissues and shunting it into our low pressure venous system, because this line is low pressure going back to the heart. That way it can be reabsorbed later down the line. And there are little filters. We'll talk about more about this in a second, but the lymph nodes are places where immune cells can actually be exposed to pathogens and sort of prepare the body for some type of event. So the structure of a blood vessel wall. As mentioned, there are different anatomies of vasculature depending on what your actual job in the system is. And so we'll start off with the lumen. Anytime you hear the word lumen, it just means a space surrounded by a wall, usually a membrane. In this case, the lumen is the actual blood conducting space. And so most blood vessels are going to have 3 layers. In anatomy, we call these layers tunics. You'll see this other places as well, for instance, the eye. But in the blood vessel, it is the intima, media, and externa. So tunica intima, tunica media, and tunica externa. The capillaries are much thinner. They're only a single layer of cell thick, and that just allows for diffusion of gas and nutrients to those tissues that are hungry for that stuff. And so this layer is actually composed of an endothelium. But moving on, you have the tunica externa on the outside, the tunica media in the middle, and then the tunica intima is actually in contact with the blood. So innermost layer, intimate contact with the blood. It's 1 or 2 cells thick, and it's an endothelium as well. In this case, a simple squamous epithelium lining that lumen. So simple squamous, so a single layer of these sort of flattened epithelial cells. Moving on, you have the tunica media. This is the middle layer. This is where you're going to find smooth muscle, but also lots of elastin. And so the ability to contract, but also the ability to flex. And so this gives us vasoconstriction and vasodilation. And by modulating that lumen diameter, what you're actually doing is modulating blood flow and blood pressure. So the third layer then, the tunica externa, also called the adventitia. This is the outermost layer. It has thick collagen fibers, essentially providing protection and support against the blood flow and that immense pressure coming off of that initial pump. So this is where you're going to get innervation by nerve fibers. This is where the lymphatic system is actually going to merge in on that venous side. And depending on how large the vessel is itself, it might actually require its own blood supply. When that occurs, we call that a vasa vasorum. It is a blood supply for the outer layer of a blood vessel. So you have the tunica intima, innermost layer. It's a couple layers thick of endothelium. We've got subendothelial layers, an internal elastic membrane. Moving out to that middle muscular layer, the tunica media. That is smooth muscle, so it's involuntary, and elastic, so it can flex. And then you have this sort of rigid, collagenous tunica externa, sometimes called tunica adventitia, and depending on how large it is, it may actually have its own blood supply. So first, let's look at arteries specifically. So we're looking at going away from the heart. There are three main types of arteries. They can be elastic, muscular, or arterioles. And so the thickness of the layers of tissue will change depending on the job that this type of artery is supposed to do. So if you look at the elastic artery, thin tunica externa, thick tunica media, and then your tunica intima is going to be about the same, I think. But the muscular artery, tunica externa, is getting a little bit larger in order to help resist the forces generated here, but the tunica media, a little bit smaller than this, and then your tunica intima again. Then finally your tunica externa, tunica media, tunica intima. But let's go through these artery types on their own. So elastic arteries are going to be something like the aorta. They are thick-walled. They have a very large lumen, so allowing for a lot of blood to move through at once. And you have elastin located in all three layers. And so these also have lots of smooth muscle, but they can't vasoconstrict. So it's going to be different from the muscular layers when we actually get there, even though it still has that same type of tissue. The job of elastic arteries is to expand and recoil in such a way that you are smoothing out the blood pressure off of that initial pump from the heart. And so the heart itself is producing a high pressure wave of blood. As it hits that joint in the aorta, that sort of curve, it's going to dissipate that pressure by absorbing the force with the elastin and allowing it to flex. And basically, as it moves through the joint there, it will leave the artery at a consistent pressure from there on. So from the elastic arteries, you go to the muscular arteries. These have a medium width lumen, so we're sort of squeezing the blood down, have the thickest tunica media, but also have lots of smooth muscle. And this is also where vasoconstriction and vasodilation are taking place, but you have less elastic tissue in this layer. So when you look at it in cross-section, the lumen is starting to become somewhat medium, but your tunica media is thick. And unlike in the elastic arteries, now you've lost that elastic protein, elastin. So continuing on to the smallest of the arteries, we have the arteriole. And so there are two types, really large arterioles. You will see that characteristic three tunics. If it's a small arteriole, it will only have a media and an intima. It loses that externa. And so the job of an arteriole really is to modulate blood pressure moving into a capillary. And it does this by vasodilation and vasoconstriction. Remember, the capillaries are only one cell layer thick. So as mentioned, you need to control the blood pressure going through them or else you'll just burst it. So it has the narrowest of the lumens. It has very little elastin and a very thin tunica externa. And in fact, the smaller arterioles are going to lack it entirely. So moving on to the capillaries. Capillaries are actually microscopic. The lumen is so narrow that a single red blood cell can pass through at a time. And because of this, it's only going to need really that tunica intima, and usually that layer is only going to be one cell thick. Capillaries are supplying essentially all tissues and cells. There are some avascular tissues like cartilage, the epithelial, which has connecting tissue supporting it, the cornea and the lens of the eye. All other cell types and tissues are getting supplied via this means. So the capillaries contain their own unique stem cell type. We call these parasites, not like a parasite like that needs a host to reproduce, but parasite is a stem cell that repairs damaged capillaries. As mentioned, because they're only a cell thick, they are capable of being damaged by simple changes in blood pressure, and so you need these parasites really to repair the damage done. And so the function of a capillary then is the exchange of gases and nutrients, but also waste on the way back, and also transmitting hormones. Remember, the endocrine system is using the blood vasculature to deliver hormones around the body. And so this is going to exchange then via diffusion between blood and the interstitial fluids. Now you have different types of capillaries to do different types of jobs. All capillary endothelial cells are joined by a special type of junction called a tight junction, but they also have gap junctions, which, remember, allow for the passage of materials. And so these are in the intercellular clefts, but basically allowing for exchange of fluids and the smaller solutes. They're gonna block the big ones, but smaller solutes can continue to pass. And so you have continuous capillaries, fenestrated capillaries, and sinusoidal capillaries. So whenever you see the word fenestrated, that means it usually has perforations or pores that allow things to pass through if they're a certain side. And remember, a sinusoid, is where things can drain. And so in a continuous capillary, the tight junctions are holding these adjacent cells together in such a way that only small gases can move in and out easily. In a fenestrated capillary, the capillary is perforated by these small holes, these pores, and so the liquid portion of the blood can move through, but not the blood cells themselves. They're too big. Then in the sinusoidal cavity, now you have these large holes in the vessel walls, and even the blood cells can enter and exit. So continuous is the most abundant, but really the focus is, of course, gas exchange at this level. So that makes sense. But the fenestrated, this is going to be found in areas where blood plasma is exchanged. So think of the great filters that we're sort of talking about, like the kidneys, for instance, which filter the blood, the intestines, and places where endocrine hormone secretion is taking place. So it looks like Swiss cheese because it has pores called fenestration. Then you have the sinusoidal capillaries, also fenestrated, but now you're having these larger intercellular clefts where large molecules and blood cells can actually pass through. So the blood flow is going to be a little sluggish in these areas, but again found only in the liver, bone marrow, spleen, and adrenal medulla. So capillaries sort of converge into a capillary bed, and the bed itself is connecting the arterial side and the venual side. So the arterial side is delivering the oxygenated blood. The venual side is draining the deoxygenated blood and any type of metabolic waste it might have picked up. A terminal arteriole is the branch off the arteriole actually connecting to the bed. And through this terminal arteriole, the diameter is going to control the flow rate and flow pressure. And so they can dilate when blood is needed, and they can shunt blood away when not needed. So there is a level of regulation here based on the need for gas and nutrient exchange. There are special capillary beds located in the intestinal mesenteries, and they have two features that allow them to control blood flow here. The first being the precapillary sphincter. So before it reaches the capillary, there is a sphincter that can regulate blood flow into the capillary bed. On the venual side, there is a vascular shunt. So essentially connecting the arteriole with the venule, bypassing the capillary bed altogether. And you might ask yourself, well, why would you want to do that? And the answer is, what about during a sympathetic nervous response? What about fight or flight? Is it really important to continue to digest your food in fight or flight? Or is that blood better used going towards your muscles or your brain or your heart? And so these mesenteric capillary beds, just keep in mind they have some special features that help to control blood to the digestive system. And so that's what that looks like. You can close off the precapillary sphincters and you can shunt blood from the arteriole directly to the venule. And so some capillary transport mechanisms that would include things like the intracellular cleft with the pores, the endothelial cells that you're diffusing across, of course, tight junctions, kind of keeping those continuous capillaries together, etc. Okay, so that was arteries and capillaries. So let's move on to veins. Veins generally carry blood towards the heart. And so they start out as venules and then merge into the larger veins. The venules, of course, it's the capillary side that's already deoxygenated. They'll form the postcapillary venule. Then those larger venules will have a little bit of smooth muscle in them as well. And then you move into the larger veins. Again, different anatomy based on different needs. Here you're not squeezing when you're not having a lot of pressure really. So your tunica media is thin, your tunica externa is thick, and your lumens are very large because really the job of veins is capacitance. You're just trying to get that blood back to the heart. And sure enough, if you look at the distribution of blood, at a given time, 64% of the blood is in the veins themselves. So large lumen, thin walls, they are good storage vessels. So as mentioned before, the blood pressure is lower in the veins by necessity, and that just makes sure that it gets pushed back to the heart. So the large diameter lumen is not constricting it, there's little resistance, and you actually need these venous valves to sort of prevent backflow of the blood itself. Remember, backflow is never good, you end up having to pump it twice. And so there's this idea of a muscular pump that the skeletal muscle and that staying active actually helps to milk blood back to the heart. Okay, let's talk about blood pressure. We talked a little bit about how the kidneys control it in endocrine with the angiotensin and renin systems, but talking about blood pressure, we're really talking about a force per unit exerted on the wall of the blood vessel itself. So this is what the vessel is actually feeling. And we kind of use an old measurement for this, millimeters mercury. This is a way to express pressure, but there is a pressure gradient in this system that provides a driving force, essentially keeping a high and a low pressure area. So any kind of resistance will be opposition to flow, and there are a couple of sources of resistance inherent to the system. That would include the viscosity of blood itself. So blood, of course, has many thousands of proteins in it. There is a hematocrit side, there's a lymph side, there's of course a serum side, and those proteins are providing some resistance to flow via viscosity. The length of the blood vessel also matters, and so does the lumen side, as we saw on the arterial side, but all of these functions are providing some resistance, causing friction between the blood and the vessel walls. So viscosity, that just refers to a stickiness, the thickness of the fluid. That's due to the formed elements, your red blood cells, white blood cells, platelets, and then any type of serum soluble plasma protein. One of those would be human serum albumin. If there's increased viscosity, you have increased resistance to flow. Obviously, the longer the vessel, the greater the resistance encountered. The blood vessel diameter itself, this is why vasoconstriction and vasodilation actually work. Depending on how big the lumen is, you can influence resistance. If the radius increases, resistance decreases, and vice versa. And so this is actually how, of course, arterioles determine the blood pressure moving into the capillary. A good example of this, try drinking a milkshake with 2 different straws. Which straw do you think it's going to be harder to actually draw the milkshake up into? Well, your vasculature follows those same rules. There is a relationship between flow, pressure, and resistance that still applies in our vasculature. And so blood flow itself, given by variable r here, is proportional to the blood pressure gradient, delta P. If delta P increases, blood flow will speed up. Blood flow is inversely proportional to resistance, given by R. So if R increases, blood flow decreases. Makes sense, right? You increase your resistance, your blood flow will decrease. You increase your pressure, blood flow will speed up. So R is actually more important in influencing local blood flow because you can simply change the blood vessel diameter and get more or less resistance. And so your systemic pressure for that systemic circuit, right, coming off the left ventricle, feeding the body, it's highest in the aorta. And really, that makes sense because it's right after that initial left ventricle pump, but it's the aorta's job to sort of absorb that and smooth it out, and so it very quickly declines afterward. So talking about arterial blood pressure, you have the systolic side. This is the pressure in the aorta during ventricular contraction. In a normal adult, it is 120 millimeters mercury. And then you have the diastolic pressure, which is the lowest level of aortic pressure when the heart is at rest. So 1 cardiac cycle, right? We've contracted, now we've rested. That is your blood pressure, systolic over diastolic. So 120 over 70 is pretty normal. The difference between these is your pulse pressure. And then, of course, a pulse, you can actually feel this on yourself. This is the throbbing of the arteries due to the difference in those pulse pressures. And of course, there's many places on the body where you can palpate that. So where most easily palpated, the temporal artery, of course, on the head, the facial artery, the carotid, the brachial, the radial, the femoral, the popliteal, the tibial, and the pedis. So there are also auscultatory methods, which is body sounds essentially. So your doctor can use a stethoscope to listen to the sound of blood actually entering into the artery. This would be done with a sphygmomanometer. We call these Korotkoff sounds, sounds of the heart essentially. So a systolic pressure normally less than 120. And so this pressure is when the sound first occurs as blood starts to spurt through the artery. So that is what you're actually hearing. And then the diastolic, normally less than 80, the sound will disappear because your artery is no longer constricted and the blood is flowing freely. So auscultatory methods, listen to sounds. Korotkoff sounds are sounds of the heart pumping blood into the vasculature. So how do you regulate blood pressure? Obviously this is important because you can damage the vasculature, you can destroy capillaries if it's too high. You need the cooperation of the heart, the blood vessels, and surprisingly the kidneys as well, and then it's all sort of integrated and supervised by the brain. So short-term regulation is due to neural controls, hormonal controls, and then finally in long-term you have renal controls. So let's talk about the neural controls. So we have this idea of a mean arterial pressure. This is just an average, right? This is the average arterial pressure throughout one cardiac cycle. So systole and diastole. The mechanisms that control resistance in this area would be, of course, the blood vessel diameter. And this can alter the blood distribution to organs in response to specific demands. And so this would be, again, fight or flight, rest and digest. Some reflex arcs that are involved in this. The cardiovascular center and the vasomotor center in the medulla, again located together. Not that strange really, medulla oblongata hosts a lot of these sort of primordial mechanisms. You also have baroreceptors. So baroreceptors are sensing changes in pressure. Chemoreceptors Perhaps there are ligands that are setting off certain receptors that say that the blood pressure is too high. And then higher brain centers are sort of integrating this information as well. So the cardiovascular center, these are going to be composed of sympathetic neurons in the medulla. And these have the cardioinhibitory and cardioacceleratory centers that we talked about in topic 2, and the vasomotor center, which is located nearby. The vasomotor center is sending impulses via the efferent sympathetic neurons called the vasomotor fibers to the blood vessels. And so this is where we get our tone from. Tone is this idea that the muscles need to be somewhat constricted sort of passively. And so your vasomotor tone then is maintained by the vasomotor center. And so it's integrating input from the baroreceptors, chemoreceptors, as well as the higher brain centers. So let's talk about the baroreceptors then. So they are sensing changes in blood volume by blood pressure. And so if your mean arterial pressure is low, you will lead to vasoconstriction. And it does this by stimulating the vasomotor and cardiovascular brain centers, resulting in an increased blood pressure, right? You squeeze that lumen and reduce its diameter, you have increased the blood pressure. Inversely, if your mean arterial pressure is too high, you need to open up, you need to dilate that lumen, and it does this by inhibiting the vasomotor center and the cardiovascular brain centers. And so by increasing the size of the diameter of the lumen, you have decreased your blood pressure. So continuing on with the chemoreceptor reflexes, these are going to control respiration, cardiac output, and regional blood flow. Basically, they want to make sure that the important organs, like the brain and the heart, are getting as much oxygen as necessary. So the aortic arch and the larger arteries located in the neck have chemoreceptors that are modulating and measuring CO2, and they can do this by measuring a drop in pH, for instance, or a drop in oxygen. And so this causes increased blood pressure by signaling those centers in the brain, like the cardioacceleratory center, to increase carbon monoxide. This will signal the vasomotor center to increase vasoconstriction, which of course will increase blood pressure. Moving on to hormonal controls. So hormones can regulate blood pressure in the short term by changes in blood volume and changes in resistance. And so remember, endocrine mediating the hormonal response. In this case, it is the adrenal medulla hormones, the catecholamines, epinephrine, and norepinephrine. And so these come from the medulla of the adrenal gland. They'll increase carbon monoxide and vasoconstriction. So these are our sympathetic, parasympathetic responders, basically fight or flight, rest and digest. Angiotensin II is stimulating vasoconstriction. Antidiuretic hormones in high levels can cause vasoconstriction. Remember, these are mostly responsible for modulating our fluid level, so that makes sense. And then you have the atrial natriuretic peptide, which is actually in the heart itself, decreases blood pressure by antagonizing aldosterone, ultimately leading to a decrease in the blood volume. So long-term renal mechanisms. So these will be modulated by the kidneys, the renal system. These are going to work by altering blood volume as well. There is the direct renal mechanism, where you simply eliminate more urine, and in this case, or possibly even conserve water to go the other way. If you need to decrease blood pressure, you can pee more. If you need to increase blood pressure, you can pee less. So that is the direct renal mechanism. Then there is an indirect renal mechanism through a renin-angiotensin-aldosterone hormonal pathway. And so going into that, this mechanism decreases the arterial blood pressure after release of renin from the kidneys. So what happens? The renin is an enzyme that catalyzes the conversion of angiotensinogen from the liver to angiotensin 1. Then you have an ACE, an angiotensin converting enzyme, from the lungs, converting angiotensin 1 to angiotensin 2. There are a couple ways that angiotensin 2 is actually stabilizing the blood pressure, and the extracellular fluid levels. The first of those is aldosterone secretion. The second is antidiuretic release. This will be from the posterior pituitary. Remember, the posterior pituitary is not actually synthesizing hormones. It is storing those synthesized from the hypothalamus. It can trigger the hypothalamic thirst center, so telling the body it needs to drink more water. And it also is a potent vasoconstrictor itself, so it can directly increase blood pressure by vasoconstricting. And so long-term blood pressure control then isn't focused on resistance so much as changing the blood volume. And how do you do that? By modulating the amount of fluid that actually enters into this system. And so that's what this figure is showing here. Click 20 times to get through this figure. There we go. Okay, moving on to the lymphatic system. So there are a couple of organs associated with the lymphatic system. That would include the spleen, thymus, tonsils, lymph nodes, and there will be some other lymph tissues that we talk about. But the lymphatic system is essentially a series of lymphatic vessels that we call the lymphatics that conduct lymph, which is fluid, to the vessels of the veins. And along the way, they're going to pass through lymph nodes, which will actually help to clean the lymph before that lymphatic fluid gets back to the heart. And so this is actually the structural basis of the immune system. I might have made a comment before about organ systems. We don't really count the immune system as its own system because the immune system is really cellularly mediated. Really, the lymphatic is as close as we get. But moving on. So we want this flow to also only go one way. It's a one-way system. Very similar to the blood capillaries, but in this case actually more permeable. That makes sense because we're trying to drain these tissues of the sort of waste products and excess fluid that's there. And so the lymphatic system can actually take up larger molecules and particles that normal blood capillaries cannot, even though the blood capillaries are also doing something very similar. So for example, proteins, cellular debris, pathogens, and even cancer cells. So this is also a way for pathogens and cancer cells to travel throughout the body. And so this is why we need our lymph nodes to help us kind of catch those. So there are specialized structures in the lymphatics that help to increase its permeability. The first will be the mini valves formed from overlapping endothelial cells. They form a one-way valve that allows for the passage of materials. Lymph can flow into the lymph capillaries but not out of them. If you have a high ECF volume, the valves will open up even more, and thus more fluid will be drained from the lymph and your ECF volume will decrease. Decreases will cause those valves to close, and so this is how the system works. Any type of positive pressure is pushing fluid into the lymph to be filtered and sent back to the heart. Once it reaches a certain level, the valves close, and thus you constantly have a fluid level in your tissues. So just like veins, you have smaller lymphs sort of leading to bigger lymph vessels. They drain into increasingly larger vessels called collecting vessels. And these collecting vessels lead to the trunks, which is a unit of the larger collecting vessels, basically draining large areas of the body. There are two large lymphatic ducts, and it's actually kind of strange how asymmetric this system is. But the right lymphatic duct is draining the right upper arm and the right side of the head and the thorax. The thoracic duct, or left lymphatic duct, is draining the entire rest of the body. So it's kind of strange. About 25% of the body by mass is being drained by the right lymphatic duct, the rest by the thoracic duct. So these are actually pumping into venous circulation, and so other forces are causing the pressure that will force them into the low pressure venous circulation. And this is at the junction of the internal jugular and the subclavian veins depending on the side of the body. And So here's that sort of weird asymmetry I was talking about earlier. Everything sort of above and to the right is being drained by the right lymphatic duct. Everything else on the left and below is being drained by the thoracic duct. So this system is also very low pressure, like the venous system. Obviously, it has to be high enough pressure to get in to the venous side, but it's propelled by some of the same mechanisms that's pushing blood towards the heart. One would be the milking action, of course, of the skeletal muscle as you work out and move around. You've got valves in the lymphatic system that are preventing backflow. You've got the nearby arteries also pulsing as they, of course, vasoconstrict and vasodilate. And then the lymphatics themselves also have a layer of smooth muscle. So any type of physical activity is actually increasing flow of lymph. And so this is part of the reason why if you're bedridden for a long time, your tissues will start to swell. So let's look at lymphoid cell types. So these are going to consist of the immune system cells and the supporting cells forming the lymphatics tissue structure. So the first of these will be the lymphocytes, of which there are two kinds. Really, we'll talk about a third when it comes to immunity, but the two that we're talking about today are part of the adaptive immune system, and they will mature into two main types: the T lymphocyte, which matures in the thymus, and the B lymphocyte, which matures in the bone marrow. And then, of course, the supporting cells, such as the smooth muscle we talked about that can help pulse and push that lymph fluid. So T and B cells' job is to protect us against antigens. Antigens are non-self. They're anything the body's going to recognize as something that should not be here. And so examples of this include pathogenic bacteria, toxins, viruses. If something is wrong with like a non-healthy cell, even cancer cells. T cells are sort of our big mediator of immune response. They can directly attack cells, and they can directly destroy infected cells, such as cancer cells or virus-infected cells. B cells, on the other hand, are mediating another type of response. Basically, they will differentiate into a plasma cell. The plasma cell will then secrete antibodies. Antibodies are these protein-based units that will go out and tag for recognition or opsonization, and basically say this is something that needs to be destroyed, in this case by phagocytosis or some other mean. So moving on, we have other lymphoid immune cells like macrophages. Macrophages help to activate T cells, but they're also out there doing some cleanup themselves. They're phagocytizing it, so it is a form of active transport, but cellular debris, pathogens, etc. And then you have dendritic cells. Anytime you hear the word dendritic or dendrite, it means tree-like, and so you should picture lots of projections coming off this cell. And so it will actually capture antigens and deliver them to the lymph node. That way, it can be presented essentially to the lymphocytes. Some supporting lymphoid cells. So you have the reticular cells. Reticular cells, as the name implies, produce reticular fibers. These fibers make up the stroma in the lymphoid organs, so sort of that scaffolding that the immune cells kind of hang out on. You can see that here. We've got some macrophages patrolling the reticular connective tissue of a human lymph node. The reticular cells are what synthesize those fibers. And then you have the lymphocytes and macrophages sort of on patrol in these areas, waiting for either activation or an encounter with some type of pathogen. So let's talk about these larger lymphoid tissues. So we just finished cell types. Let's talk about larger organizations of cell types. Well, the job of the lymphoid tissue is basically to provide a site for lymphocytes to actually get activated and proliferate. And so these are areas of surveillance, essentially, for the lymphocytes and macrophages as they filter through that sort of excess lymph fluid. By mass, it's mostly this reticular connective tissue, which is a type of loose connective tissue. I mean, you can palpate the nodes in your neck, and you can kind of feel like it's a loose kind of sac. But the macrophages will live on those reticular fibers, and then the spaces between them offer a lymphocyte a place to stay when it's patrolling. And so it's essentially like a checkpoint. There are different types of lymphoid organs, including the lymph nodes, which are more solid, but there are also sort of these diffuse lymphoid tissues as well. The lymph nodes themselves are solid spherical bodies, tightly packed lymphoid cells and reticular fibers. Again, you can palpate some like in the nodes of your neck. Interestingly, kind of asymmetric. The asymmetric nature of the lymph nodes is actually exaggerated in males a little bit. But these lymph nodes contain the germinal centers of the proliferating B cells. And so B cells need to go through two different types of selection, a positive and a negative selection, to make sure that they will actually respond when a threat is encountered and to make sure that they're not reacting to self. And so in the appendix, we used to think that perhaps the appendix was vestigial. It seems now that there's some different hypotheses on what the appendix actually does. But the appendix actually does have some lymph tissue there, the Peyer's patches. So perhaps it is involved in some way in the immune response. Then in diffuse lymphoid tissue, this is where you're going to find a loose arrangement of lymphoid cells and reticular fibers found virtually in every body organ. So every body organ essentially is going to have an area where this sort of fluid can be monitored by macrophages and lymphocytes. Now there is two tiers to this system. There's the primary lymphoid organs and the secondary lymphoid organs. The primary lymphoid organs are simply where lymphocytes mature. So really there's only two of those, red bone marrow and the thymus. Remember, both originate in the bone marrow, and that's where the B cells will mature, but T cells will migrate to the thymus and mature here. And again, the thymus is kind of interesting because in animals, the thymus will actually deteriorate and atrophy with age. And so this is part of the reason why old people probably get sick more. Now, the secondary lymphoid organs are areas where those mature lymphocytes will be sent out to actually encounter and become induced. And so that's going to be the lymph nodes, the spleen, the mucosa-associated lymphoid tissue, and those diffuse lymphoid tissues, which are located essentially in every organ. So this is what that looks like. Primary, you have your thymus, which is just superior to the heart. Then you have the red bone marrow, which of course is where the lymphocytes are made, but then the T cells will migrate to the thymus. Then secondary organs, you have the lymph nodes, the tonsils, the spleen, the Peyer's patches, and the appendix. So what is the job of a lymph node then? Well, it's actually filtering the lymph. For instance, the macrophages there are destroying any type of microorganism they recognize and any type of cellular debris that has entered the lymph. Basically, you don't want that cellular debris ending up in the blood. There are a couple reasons for that, but immune system activation as well, allowing the lymphocytes a chance to become activated and mount an attack against antigens. Because remember, these lymphocytes, they have kind of an off form where they're not doing anything. They actually have to be shown what antigen they're meant to attack. And so the lymph nodes are a place where that can happen. So, talking about our secondary organs, you have the spleen. The spleen is a blood-rich organ, about the size of a fist. It's actually kind of long-shaped. It's on the left side of the abdominal cavity, just below the stomach. This is our largest lymphoid organ, and it is the site of lymphocyte proliferation and immune surveillance. But it's also cleaning the blood, essentially. The spleen is a filter. It's removing aged blood cells and platelets, which are another formed element. And there are these macrophages in the spleen, the lymph tissue, actually doing that. So keep that in mind. Then you have the tonsils. The tonsils are the simplest lymphoid organs, and they are the filters for our sort of oral and nasal routes, right? You're breathing in air from the atmosphere, the air is dirty. You're eating food, perhaps the food is not clean. The tonsils allow for some exposure via the lymph tissue there to induce the lymphocytes that are located there. So Peyer's patches, these are going to be on the distal portion of the small intestine. Of course, we associate them with the appendix as well. And so its function is to destroy bacteria, basically preventing them from breaching the intestinal wall. The intestinal wall is controlled very tightly by a sort of tight barrier, and so these Peyer's patches are allowing a line of defense against the bacteria that are there, but also generate memory lymphocytes that can help to remember specific threats encountered before. And so this time, the next time that threat is encountered, a quicker response can be mounted. Then you have the appendix. When you look at the appendix, it's actually just a kind of a sac hanging off the large intestine. And its location is like the Peyer's patches to prevent bacteria from breaching the intestinal wall and to generate memory lymphocytes. Now, the appendix probably had multiple jobs in the past. We used to think that maybe this is where we housed our bacteria that could help to ferment things like toxic leaves and vegetation, and we've even thought that this is perhaps like a reservoir for good bacteria. But this is definitely one of the jobs of the appendix. And then you have the thymus. So the thymus is located in the inferior neck. It's just above the heart. And this is where the T cells actually mature. Largest during childhood, as mentioned, and stops growing during adolescence and then atrophies. And it will still produce those immunocompetent cells, although at a slower rate. Okay, so that is the end of topic 3, blood vessels and lymphatics. Lots of information, but I believe that is all for the first module exam.